What is protein folding? Well, to understand protein folding, we need to understand the basics of protein synthesis within a cell. This is a human cell with a bunch of wacky stuff like a ribosome or like a mitochondria. But the most important is a nucleus, which holds the genetic code of the organism. The genetic code is held within the DNA of the organism in this iconic double helix. The DNA codes for a bunch of stuff like how tall you are, the color of your hair, and what color your eyes are. This part of the DNA is called a nucleotide. The structural formula for a nucleotide is pretty overwhelming when you look at it with all these shapes and different letters. You can see right here there's a phosphate group right here, a sugar group, and a nitrogen base. An easier way to visualize this is with the pool house garage model, where you have the circle being the phosphate group, the pentagon being the sugar group, and the rectangle being the nitrogen base. In DNA, the nitrogen bases are adenine, cytosine, guanine, and thymine. Each base has its own specific shape. Guanine into cytosine and thymine into adenine. You cannot put thymine into cytosine or the other way around. The one big problem with DNA is that it can't get outside the nucleus. So it needs to find another way to send its genetic code. This is where RNA comes in. The structure of RNA is very similar to DNA, but it has a different sugar group and a different nitrogen base named uracil. There are multiple types of RNA. The first is mRNA, that takes the genetic material in the DNA and brings it out of the nucleus. During the duplication of DNA, the mRNA goes in and takes the code and matches it with the opposite nitrogen base, with thymine going to adenine and adenine going to uracil, because RNA does not have thymine. When it leaves the nucleus, it heads towards the ribosome for the next step of protein synthesis. Before heading into the ribosome, a term we need to know is codon, which is the title given to a group of 3 bases next to each other. Inside the ribosome, the mRNA meets with the tRNA that takes each codon of the mRNA and pairs it with its anticodon and makes an amino acid. There is a chart to help with finding the amino acid with each anticodon. This chart has over 64 different codons. So for this combination, I would look for A, U, and U to get isoleucine, and then tRNA would make the amino acid. Here is an example of tRNA reading different codons and making an amino acid chain. Amino acids are the building blocks for protein. Amino acids are special because the change of one part can cause it to have different characteristics in a protein. For example, adding a hydrogen makes one glycine, but adding a carbon, 2 hydrogen, and a benzene ring makes phenylalanine. So, an amino acid chain is also a polypeptide chain due to its peptide bonds, or simply called a protein chain. For the primary structure of protein folding, you simply have just one protein. The secondary structure is either a alpha helix or a beta sheet. The alpha helix is a spiral of protein chains with hydrogen bonds going through the middle of it. Beta sheets are 2 protein chains connected with hydrogen bonds. When the 2 chains are connected with nitrogen, and carbon, it's called a parallel structure. If it folds in on itself and nitrogen goes with carbon, it's called an anti-parallel structure. The tertiary structure makes the protein hydrophobic and sometimes form a disulfide bridge. When it is put into water, the nonpolar part of the protein goes into the middle. The polar ends wind up outside and attach to the water molecules. Disulfide bridges happen when a cytosine amino acid and a thiol group with a sulfur ion are connected and oxidize, and the formation of covalent bonds are connected by the sulfur ions. The formation of the bridge happens on the outside of the cell, and the separation is in the interior. The interior is also going through reduction, and the outside is going through oxidation. The final structure is the grouping of multiple polypeptides. Each polypeptide is given a number, and a name based on the number of polypeptides. This is called a tetramer, since it has 4. In order for this to be successful, each protein structure has to have the proper conformation. If one fails, that can cause problems and diseases to occur within the protein.